Hi, science friends. This is the last lesson in genetics, so we're going to finish up your notes today. And we are going to talk about some mutations that alter chromosome number. So it's important to know that there are a couple of type of mutations that you can have that result in genetic disorders. So today we are focusing on chromosomal mutations. We will talk more about in the next unit gene mutations. So remember, in your DNA is a sequence of A's and T's and G's and C's, okay? And so you have a sequence of A's and T's and G's and C's. So we would have a lot more DNA and a lot more DNA on either side of this if we're reading all of our DNA. This one segment, maybe, it's too short technically to code for a gene, but we're going to do an abbreviated version. This is a gene that codes for red blood cells, okay? And so somebody that has sickle cell anemia, that's a condition of the red blood cell. Some of you have maybe studied it if you took Project Lead the Way before. Their red blood cells don't form the normal shape. They have a sickle cell. They look kind of like a banana. And so that is a gene mutation. It's a mutation in one gene. And so basically what happens in a gene mutation is you have a letter that's swapped, or you get an extra letter, or you get a letter that's deleted, or a T is changed to an A or whatever. That is a gene mutation, okay? You can't see those in a karyotype. Now, you could observe them if you had like gene sequencing or you did some really in-depth genetic testing, but you can't see that in a karyotype. Today, we are talking about chromosomal mutations. And so they are larger scale mutations on a chromosome. These can be really severe. Gene mutations can be really severe, but it's affecting one small spot on the chromosome, okay? Not affecting the whole chromosome. So that's important. Gene mutations and chromosome mutations. And we'll talk more about gene mutations later. Today we're going to focus on these guys, the chromosomal mutations, okay? So one thing that can happen with chromosome is a phenomenon called non-disjunction. And so in meiosis, sometimes the chromosomes get stuck together and they don't separate properly. It can happen in meiosis 1 when the homologous chromosomes separate, like in this one, or it can happen in meiosis 2 down here when the sister chromatids separate. But what you see, regardless of when it happens, if it's in meiosis 1 or meiosis 2, is you have gametes that have the incorrect number of chromosomes, okay? So in this example here, these gametes turned out fine. They have the correct N, which is the haploid number of chromosomes. In this case, N is equal to 2, okay? And so if they joined with another normal gamete that had 2, that looked basically just like it, then they would have 2 in the diploid number of chromosomes when they joined, and that would be perfect, okay? These, though, and these over here, do not have the right number of chromosomes. This has N minus 1. It has 1 too few chromosomes. This has N plus 1. It has 1 too many chromosomes. And that is the result of non-disjunction. Those sister chromatids got stuck together. So, what phase of meiosis does this happen in? Interphase, prophase, metaphase, anaphase, telophase. What phase does this happen in? You should say anaphase, okay? This one is showing anaphase 1, and this one showing anaphase 2. So non-disjunction is failure of chromosomes to separate in anaphase 1 or 2 of meiosis, resulting in gametes that have an irregular number of chromosomes, okay? And so that's basically what this says here. Non-disjunction can be pairs of homologous chromosomes, but it could also be when the sisters don't separate too, okay? So you may want to add that in there. Can also be pairs of homologous or sisters, the sister chromatids. Okay? And so, as a result, one gamete receives 2 of the same type of chromosome, and the other doesn't see or get a copy. So this one lacked that other chromosome. This one got 2 copies of the same chromosome as a result of non-disjunction. Okay? And so the result of this is known as aneuploidy, a chromosomal aberration where one or more chromosomes are present in extra copies or deficient in number. When there's an irregular number of chromosomes, it's called aneuploidy. Okay? So non-disjunction is the process where they don't separate. That's the failure to separate. Aneuploidy is the result. An irregular number of chromosomes, 1 too many or 1 too few, is aneuploidy. Okay? So we see this in trisomy 21. Whoops. What is trisomy 21? Trisomy 21 is Down syndrome. Sure, you're all familiar with Down syndrome, but individuals with trisomy 21 have an extra 21st chromosome. They should have 2 sets of, one 21st chromosome from their mom and one 21st chromosome from their dad. But one parent, either mom or dad, had non-disjunction occur, and their gametes didn't divide properly. They didn't split apart in anaphase like they were supposed to. And as a result, either the sperm or the egg had an extra 21st chromosome. And so they resulted in trisomy 21. Okay, and they have Down syndrome. Now, some of these chromosomal aberrations can be lethal. And what I mean by that is sometimes if it's a chromosome that encodes for a lot of really important genes or genes that are just absolutely necessary, it can be lethal, meaning it can result in like a spontaneous abortion in utero. And so that child will fail to develop, essentially. Okay. Now, the next thing I want to talk about is polyploidy. Polyploidy is important in plants. We don't see polyploidy in humans, okay? But we do see polyploidy in plants. And so polyploidy is when they have a whole extra set of chromosomes, right? So for humans, our haploid number is 23, okay? Our diploid number, when you get sperm and egg together to make a real human, then it's 46. But polyploidy would be if you had like 3N or 4N or 5N. They have whole extra sets of chromosomes. And we see that a lot of time in plants. Strawberries are notorious for this. Bananas are notorious for this. Wheat is notorious for this. They'll have many sets of chromosomes. And so that's why when we do DNA extraction, if we were in class, we would do a DNA extraction. Maybe we'll do that next week when y'all are here one day. Then we often use strawberries because they have a lot of DNA, because they are polyploid. They have extra sets. And so it's easy to be able to pull that DNA out of the cells and see it with your eye because there's so much DNA present in plants. Okay? And so that's what polyploidy is. More than 2 complete sets. Not an extra one. That's aneuploidy. A whole extra set. Okay? So, another thing that can happen is an alteration in the chromosome structure. Okay? And so it's missing a big, you can have so our original chromosome, let's say, looks like this. Okay? Now, you can have a deletion. And so we see that we've deleted the B segment here. So anything that segment of chromosomes, any genes that were in that area, those are gone, okay? That B segment has been deleted. So we lost a chromosomal segment. You can have a duplication, and so you've got double for there, okay? You can have an inversion, and so parts of the chromosomes have been broken and switched places and split. And then you can have a translocation where you, with a homologous chromosome, those kind of split and separate, okay? And so moves a segment from one chromosome to another. is a translocation. And so those are some other things that you can have alterations in chromosome structures. Some of these you may be able to see in a karyotype. So for example, if you're looking at your karyotype and you see your chromosomes lined up, and this is like chromosome set number 2, and one of them's like yay long and the other one's like this long. Well, chances are, there's been an issue there. There's a duplication in this chromosome, or there's a deletion in that chromosome. So some of these alterations in chromosome structures can be seen in karyotypes. But to be able to, figure out more about the genes that were lost or duplicated or whatever, you would have to do some genetic testing that's a little bit more intense than just a karyotype. Okay? Now, the last thing, genomic imprinting. This is kind of an interesting thing. So genomic imprinting is when you have a variation in phenotype depending on whether an allele came from your mom or your dad. Okay? Now, this is not the same thing as a sex-linked trait. Okay? In genomic imprinting, let's say your mom gave you big A and your dad gave you little a. Okay? So normally you would show whatever the dominant trait is. All right? But sometimes in genomic imprinting, one allele will silence the other. Okay? So let's say that the mom's allele got silenced. That version got silenced. It doesn't get deleted, it just gets turned off for whatever reason. And so then instead of showing the dominant phenotype, then you would show the recessive phenotype. And then in your children, when you go to pass off, normally you would have a 50% chance of passing that one or a 50% chance of passing that one. But the one that got silenced will never be passed. So in your children, you would always pass the little a, okay? And so that would remain for every generation that follows. And so sometimes, it's just something that happens sometimes. So you have genomic imprinting where an allele gets turned off, and it's random as to which one it happens to. So that's a bit of an interesting phenomenon. And then the last thing I want to talk about is extranuclear genes. So extranuclear, that's going to mean outside of the nucleus. So remember, genes are always located in DNA. And we know that most of the DNA is inside of the nucleus. But there are 2 places outside of the nucleus that also contain DNA. Where are they? Well, in humans, there's only one, or in animals for that matter, and that is in the mitochondria. The mitochondria is one place besides the nucleus where you have DNA. And then the other place in plants, they also have a mitochondria, remember, because they do cellular respiration too. But in addition, they also have the chloroplast. So the chloroplast, the mitochondria, and the nucleus is where you find the DNA. The bulk of it being in the nucleus. So extranuclear genes, again, are genes outside of the nucleus. So in humans, mitochondrial DNA is inherited from only one parent. Any genes that are encoded on the mitochondria, you get from your mom. Okay? Mitochondrial DNA is always from your mom. So you're always going to have just a little bit more genetics, a little bit more genes from your mother than from your father. Now, the reason for that is when the sperm fertilizes the egg, the cytoplasm of the egg, which is where the mitochondria is in the cytoplasm, comes from the mom, okay? And so that's why you get the mitochondrial genes from your mom. So there is DNA there. It does code for a few things, but if you have any type of mitochondrial issues, those would have been inherited from the mother, okay? And then in plants, there's DNA in the chloroplasts as well, okay? And those are considered extranuclear genes. So you have a quick assignment today in Google Slides. Basically, you're going to be looking at some karyotypes, and you have to be able to identify any issues that are going on in them. So there are none of these. There are no chromosomal structure issues. I'm not asking you to look really, really close and say, is this one like a millimeter longer than that one? There's none of that. What I'm wanting you to look for is sex chromosomes to be able to tell me if it's an XX, it's a female, if it's an XY, it's a male. And then also look at if there's any trisomies. Do you have any extra copies of chromosomes and what chromosome number that's on? And then there are a few sex chromosome disorders. So sometimes females will inherit an X, but only one X. Or sometimes males can inherit two X's and a Y. So be on the lookout for those. And then I also ask you to just give a very brief description. I'm talking like 1 or 2 sentences, nothing major, but about what that condition is or what it's called and what kind of it causes. Okay? So that's your assignment for today. If you have any questions, let me know. And if not, then you all have a great day.